Polymerase chain reaction is something that we can use to create lots of copies of a particular segment of DNA in order to be able to use them for a number of different reasons. So if we consider, for example, a crime scene, and maybe a very, very small sample of DNA has been retrieved from a crime scene, from blood or hair or something like that, and we want to create lots and lots of copies of that DNA so we can run lots of different tests and we can observe it and sort of play around with it in a number of different ways. So in order to get all of those copies of DNA to be able to mess around with and experiment with, we can put that original small segment of DNA through the polymerase chain reaction to get lots and lots of copies. So if we start off with, so for example, we've got an initial segment of DNA. So this is our double helix, or two strands of DNA there. What's first going to happen is this DNA will be put through the denaturing stage. So this DNA is going to be heated to 95 degrees, not 95%, going to be heated to 95 degrees Celsius. So we heat our DNA up to 95 degrees Celsius, and what's going to happen is this will cause the double helix to break apart. So those bonds, those hydrogen bonds will break apart, and that double helix will now separate into its separate strands like so. Alright, so our hydrogen bonds are broken. The second step of polymerase chain reaction is called annealing. Now in annealing, what we do is we cool our DNA samples back down to 60 degrees. So something called DNA primers are able to bind to exposed bases. So DNA primers are these segments of DNA, these small segments of DNA which will come along and they might bind to, in a complementary fashion, and bind to the exposed bases, because that DNA double helix is split into two separate strands. So after this stage, we then have a third and final stage, which is extension. So now we heat the DNA back up to 72 degrees, and what happens as a result of this is nucleotides are able to bind to bases using something called Taq DNA polymerase, or with the help of something called Taq DNA polymerase. So these free nucleotides that are added to our DNA sample, they are able to go along like so and fill in the rest of the double helix, and we've now got a new copy. We've got two copies of our original DNA segment. This Taq DNA polymerase, what is special about it and different about it from a normal DNA polymerase is it's able to function and remain active at higher temperatures, like 72 degrees. So if the DNA polymerase was unable to function at those temperatures, it would obviously not be able to join the nucleotides to the exposed bases and create that double helix. So we need this special Taq DNA polymerase which is able to withstand higher temperatures. So what we would call this is like a cycle. So this is like cycle 1, for example. So then what's going to happen is we repeat the process. So we go back to step 1, we go back to the denaturing phase, and through annealing, through extension. So what will occur is this newly made double helix will be broken apart in the denaturing phase. So I have a strand up here, we have this green strand here, put in these purple strands, like so. So they will go through the denaturing stage, we get those new double helix being broken apart, the hydrogen bonds broken apart, we then get our cooling back down to 60 degrees, so we get these segments called primers joining to our exposed bases, our exposed strands, and then finally we get our extension, the reheating to 72 degrees, nucleotides, free nucleotides that we've added bind with the help of Taq DNA polymerase, which also obviously has to be added to our DNA samples that we're trying to replicate, and then those free nucleotides are going to come along and fill in the rest of that double helix. So you can see that in the course of a couple of cycles, we've gone from having one double helix to 2 copies of that double helix to now having 4 copies of that double helix. So we will get a doubling of that DNA each time, and then it only is going to take several cycles before we start to be in the hundreds and then the thousands of copies of DNA. And then these samples can be used for a number of different reasons, because we've got lots and lots of copies of an initially small amount of DNA. So that's how polymerase chain reaction can work.